In this section of our Staphylococcus lecture, we're going to focus on the methods of identification of all of the various Staphylococcus species. We already talked about some of the morphological characteristics which are key for Staphylococcus. When you see a gram-positive cocci in clusters, you're automatically going to have a light bulb that goes on that says, "Oop, Staphylococcus." We already mentioned that Staphylococcus is not motile, which means doesn't have any flagellum. It is not a spore former. It is not fussy, so it will grow on any type of nutrient agar, any supportive media. And the clinical laboratory blood agar, 5% sheep blood agar, is the commonly used general media because it does allow you to differentiate organisms that are hemolytic from those that aren't. And most bacteria, all non-fastidious bacteria, and also a couple of fastidious bacteria, like to grow on blood agar. It is a facultative organism, and we already mentioned that Staphylococcus aureus is beta hemolytic. This is a gram stain of a sputum specimen, so you can see the nice gram-positive cocci, either singles, paired up, or in small little clusters. And you can see the sputum sample, some cells in the background, some debris, which is very common for a clinical specimen. In this slide, you can see intracellular Staphylococcus organisms, the nice clustered gram-positive cocci inside white blood cells. This is a direct smear examination, so in a microscopic direct smear of a patient specimen, if you saw gram-positive cocci that were in pairs and clusters, that of course would be indicative of a staph. And a lot of times if you see a lot of what's called PMNs or polymorphonuclear cells, granulocytes, you are automatically going to think bacterial infection. So a ton of white blood cells, specifically the PMNs, is indicative of bacteria. A ton of lymphocytes usually is more indicative of a viral infection, and we're going to talk about viruses later on in the semester. This is a gram-stained smear of pus. Again, you could see the gram-positive cocci. They look a little pinkish, but they are gram-positive, and you could see white blood cells they're clinging to and some of that pus debris. This is an electron micrograph. You can see the nice circular morphology of Staphylococcus. On blood agar plates, the organism is smooth, a whitish to yellowish color, and a bit creamy looking. So the coagulase negative staph tend to be more of a whitish color. Staphylococcus aureus tends to get a golden or yellowish hue to it. Also, your Staphylococcus aureus is usually beta hemolytic, so you're going to see a little clearing around the colony on a blood agar plate. Here is coagulase negative staph on a blood agar plate. Like I said, it's more of a white color, very creamy looking, small to medium sized. Usually your CNS, coagulase negative staph, is not hemolytic. Staph saprophyticus may have a yellowish pigment, but it usually is non-hemolytic, whereas Staph aureus has more of a golden pigment and it is beta hemolytic. So usually on blood agar plates, an experienced clinical microbiologist will be able to tell the difference just by looking at the colony morphology, pretty much almost exactly what organism they're looking at. So when you go to your clinical laboratories, those of you that are not MLTs or have not been working in a clinical lab, when you start your micro rotations, really pay attention to the individuals that work there because they have a wealth of knowledge and they're going to be able to teach you so many things. Here is Staphylococcus saprophyticus on the top, or in A, and Staphylococcus epidermidis in B. So you can see that even though they're both coagulase negative staph organisms, they both look slightly different on sheep blood agar. The biochemical tests that are used to differentiate the Staphylococci are Catalase. All Staphylococcus organisms are Catalase positive. That's a key feature because that is the feature that differentiates the Staphylococcus from the Streptococcus that we're going to talk about next. You can do oxidative fermentative tests, which is oxidation and fermentation of carbohydrates. It's a test using OF media. And Staphylococcus is an oxidizer and a fermenter. So in OF media, it is going to turn the media from green to yellow. Only Staphylococcus aureus is able to ferment mannitol, so you would see that on a mannitol salt agar plate. Only Staphylococcus aureus is coagulase positive, and you can also do Novobiocin susceptibility. We're going to talk about each one of these key tests in the next several slides. So the key identification, you're in the clinical lab, you have a patient specimen, you gram stain it, and you see gram-positive cocci. You should immediately, in a clinical setting, be thinking Staphylococcus or Streptococcus. So your first step is going to do a Catalase test. So Catalase is a test where you take hydrogen peroxide, you drop it on a colony, and if it bubbles up, it means the hydrogen peroxide is getting converted to water and oxygen causing these bubbles. If you get a bubbling, then it means it's catalase positive. So for your gram-positive cocci, that's a key, because if it's catalase positive, you're automatically thinking a Staphylococcus and not a Streptococcus. So you can go to this website. I put a link on your course site page, and you can see the catalase reaction. So for those of you that have never been in a clinical microbiology lab, or if you didn't do catalase tests in your general microbiology laboratory, you should go in and watch this video. It's a very simple test. It takes 2 seconds, and it is one of the few manual tests that are actually done in the clinical laboratory. Coagulase is the test that is looking for the enzyme coagulase. There's bound coagulase and free coagulase. Bound coagulase is also called clumping factor. It's a screening test, and it converts fibrinogen to fibrin in your coagulation process. Free coagulase is a thrombokinase-like enzyme. It clots fibrinogen in the presence of plasma. So these are the 2 different types of coagulase, but the key feature of coagulase is it allows you to differentiate your Staphylococcus aureus pathogen from all of your other Staphylococcus species. There are many different types of commercial coagulase tests available. Many labs are using various tests. Again, Catalase and Coagulase are 2 of the manual tests that are actually still performed in a clinical laboratory. So Coagulase is going to detect the enzyme coagulase, and there are 2 different methods that are commonly used. One is the slide coagulase test, and the other is the tube coagulase test. More commonly, the slide test is used because these are available in kits. They're very quick. You finish them in a couple of minutes. The slide test is looking for clumping factor or bound coagulase. So on the left, you can see the slide test. On the right, you can see the tube test. On the slide test, you are mixing your organism with your reagent, and you're looking for a clumping or agglutination reaction. In the tube test, you are going to mix your organism with rabbit plasma, and if the organism produces coagulase, it's going to cause that plasma to clump together, to coagulate. So in the tube test, which is testing free coagulase, the top reaction is positive. You get a nice little clump of your plasma, and in the bottom tube test, it's still a liquid in the tube, so that's a negative coagulase test. The tube test used to be the standard coagulase test done in the clinical lab. Now the tube tests are not done anymore because you used to have to incubate the tube overnight, so you really wouldn't know if you had a Staph aureus for at least 24 hours. So with the commercial-based tests now, you can do a coagulase test in, like I said, just a minute or two. Novobiocin is another test that can be done, and it's usually performed to find Staphylococcus saprophyticus. So Staphylococcus saprophyticus is resistant. So Staph is a positive Novobiocin or a resistant reaction, and all of the other Staphylococcus species are sensitive to Novobiocin, which is a negative reaction. In the Novobiocin susceptibility tests, you would take your organism and streak it out as a lawn on a plate, and you would put a Novobiocin disc onto that plate. So you can see in this slide, on the top, that organism is resistant. It's resistant because it's able to grow right up against that disc containing Novobiocin. So that would be indicative of Staphylococcus saprophyticus. On the bottom, you have a halo around the organism where the organism couldn't grow. That means the Novobiocin inhibited the growth of the staph, which would indicate that it's not Staphylococcus saprophyticus, it's one of the other coagulase negative Staphylococci. There are other non-routine tests that will only be done in maybe a reference laboratory or depending on the specific case. So you can look for toxic shock syndrome toxin 1 production. You can do serological based tests, phage typing, probe testing. So these are not routinely done for the Staphylococci, but like I said, a reference lab or a public health lab may do some of these less routine testing. Specimen collection and handling, when specimens are taken, they need to be taken from the actual site of infection. So if there is a deep wound, the site needs to be from deep into the tissue and not a surface skin specimen. There shouldn't be a lot of delay between collection and transportation to the clinical laboratory. And I mean, in the ideal world, there would be no delay, that samples would be tested immediately after collection, but that's not really realistic. If the specimen is transported, it needs to be transported in an appropriate collection device that will prevent the organism from drying out or possibly being overgrown by contaminating organisms. So there are special transport media that are used for patient specimens. This is a flow diagram of the Staphylococcus species. So you have a gram stain and you find a gram-positive cocci. The organism is facultative. It grew in CO2, grows in ambient air. It's not fussy at all. First thing you do with a gram-positive cocci is a Catalase test. If it's Catalase negative, it would be indicative of one of the family members from the Streptococcaceae family. We're going to talk about that group of organisms next. If it is Catalase positive, it's from the Micrococcaceae family, and it would likely be indicative of a Staphylococcus organism. The next thing you want to do after a catalase and you find it's catalase positive is to do a coagulase test. If it's coagulase positive, it is Staphylococcus aureus. If it's coagulase negative, you can do an oxidase test and a bacitracin susceptibility test. So the bacitracin susceptibility test is done exactly like a Novobiocin susceptibility test, but the disc would have bacitracin on it. If the organism is oxidase positive, and oxidase is a test that's more commonly done on gram-negative organisms. But if you wanted to determine if it were a micrococcus over a staphylococcus, you would do oxidase and bacitracin. Micrococcus organisms are oxidase positive and susceptible to bacitracin. The coagulase negative staph, or CNS, are oxidase negative and resistant to bacitracin. If it were a urinary tract infection and you wanted to see if it was a Staphylococcus saprophyticus over any other coagulase negative staph, you would then do your Novobiocin susceptibility test. If it's resistant, meaning the organism grows right up against the Novobiocin disc, it's indicative of Staphylococcus saprophyticus. If it's sensitive, in other words, there's a zone around the disc, it is CNS, not Staphylococcus saprophyticus, and most likely it's a Staphylococcus epidermidis, since that's the most common coagulase negative Staphylococcus species. Prevention, same type of prevention we already talked about for Staphylococcus aureus. We're going to review a case study. A 19-year-old woman complained of fever, flank pain, difficulty urinating, feeling like she had to urinate a lot, often urgency to urinate, and had blood-tinged urine. The urinalysis revealed many white blood cells, and there were also casts in the urine that contain white blood cells. The urine culture grew 45,000 colony-forming units per mil of a white, non-hemolytic colony on blood agar, and there was no growth on MacConkey agar. So MacConkey agar is a very specific agar that is selective for gram-negative organisms. So gram-positive organisms won't grow on MacConkey agar. So if you have an organism that grows on blood but does not grow on MacConkey, that would indicate it's likely a gram-positive organism. Since it's a gram-positive organism, the very first test you want to do is a catalase test. This organism was catalase positive, so it is indicative that it's a Staphylococcus. The next thing you want to do is a coagulase test, and in this instance the coagulase test was negative, so you know it is not Staphylococcus aureus. It doesn't produce, it did not have a zone around the Novobiocin disc in the Novobiocin susceptibility test. So if there was no zone, that means that the organism grows right up against the disc. So when you read a case study like this and you start, this is the way you need to think in microbiology, just how I worded out that case. You know that this case, this infection in this individual is caused by Staphylococcus saprophyticus. Now, if this case, if you're not understanding the thought process and going through this case and coming up with the conclusion that this individual is infected with Staphylococcus saprophyticus, you might need to make a telephone appointment with me, because in microbiology, it's really important to pull everything together. So you can't just think of one small piece of it. You want to start to really think about the whole picture, the whole disease. So you will be getting case studies like this as questions on your examinations, and you're going to have to figure out what genus and species this person is infected with based on the information written up in the case. And it's really important to focus on the key information. Okay, this is a young woman, it's urine, it's gram positive, it's catalase positive, coagulase negative, so you know it's a CNS. That young woman, urine, UTI, you should automatically be thinking Staphylococcus saprophyticus, and then the Novobiocin result tells you, yes, that's what it is. So we will be going through case studies at the end of most of our lectures throughout the semester to get you thinking in this big picture way with all of these organisms. Some of the points to consider when you're thinking about a case like that is, what are the clinical manifestations? So in this case, the individual had a urinary tract infection. You want to always think about, okay, what group of organisms might cause this type of infection? So you can narrow down the organisms that you're considering. What factors might be contributing to this organism's ability to cause disease, or the virulence? So these types of questions always should be on the back of your mind when you're thinking about an organism and when we're reviewing a new genus or species of any organism.